Hello, in this video, I'm going to introduce step 1 in the workflow of measuring a binding curve experiment on the Fluidity 1M, which is sample preparation. What you will need is a fluorescently labeled protein and an unlabeled binding partner, for example, an antibody and its antigen. Typically, the smaller of the two binding partners will be labeled. The Fluidity 1M can detect fluorescence from all fluorophores that emit in the sample range, as for example, Alexa fluorophore 88, autophore 88, FTSE or GFP, and a second detection range, for example, Alexa 647, Auto 647, or Sci-Fi. Now, to prepare a binding curve experiment, we recommend using low binding plastic tubes, shown here, and low binding 96 well plates. The simplest approach for sample preparation is to first prepare an 8-point dilution series of the unlabeled protein and then add the labeled protein at a constant concentration. The reason we do this is that our microfluidic chip comes in the same format as a 96 well plate, having 3 sets of 8 ports, sample ports. So later, when it comes to loading these 8 samples on the chip, it will be very straightforward to use a multi-channel pipette and, for example, measure duplicates of an 8-point binding curve. The required concentrations depend on the binding affinity, with higher affinities requiring lower concentrations and lower affinities requiring higher concentrations to reach binding saturation. In terms of sensitivity, the Fluidity 1M can detect concentrations down to 100 picomolar for the labeled binding partner. At the higher end, protein concentrations up to millimolar range are no problem at all. As a general setup, we recommend preparing enough sample to measure in duplicate. Each sample port on the chip requires 4 microliters, so 10 microliters will be plenty. Now that we've prepared our samples, we can equilibrate them. We recommend an equilibration time of 30 minutes, but this, of course, is dependent on the interaction of interest and your protein's characteristics. During sample equilibration, the experiment can be set up on the touchscreen of the instrument, for which we have a separate video available on YouTube, LinkedIn, and our website. Thanks for watching, and let us know if this approach works well for your sample prep. If you have any additional questions, get in touch with our experts at tech support, who are always keen to help.